Hello and welcome to this week's episode of Research Goes Viral. We have a very special guest with us today, which I will introduce you in just a wee moment. My name is Stephen and I am hosting for today. I'm also joined by Anna Sims as my wonderful co-host. And today we have the absolute pleasure of Anice Lowen being with us today. So hello and welcome.
Thank you so much.
Now a little bit, I've done some research, so, as we are prone to do here at the CVR.
What a pro.
Thank you. So a little bit of background about you. So you're a current associate professor at the Department of Microbiology and Immunology at Emory University School of Medicine, which is in Atlanta, I believe. Mm-hmm. Yes. And you're also the co-principal investigator of the Emory Centre of Excellence for Influenza Research and Response. And your background, so that lab is focusing on like two aspects of fluid biology. So you're looking at transmission from, I believe it's like host-to-host transmission.
Yes.
And you're also looking at genome reassortment as well.
Yep, another major theme.
Another major theme of the lab. So you originally studied for a bachelor's in biochemistry, I believe, at the University of Alberta in Canada.
Yes, that's right.
Google didn't lead me astray. And then you actually came to a very familiar place afterwards doing your PhD. I think it was called Centre for Virus Research in Glasgow.
That's the place.
But I think it was the Church Street campus it was back in the day.
Was it called the Centre for Virus Research back then?
It actually wasn't.
Yeah.
Oh, what was it called?
I don't know.
Oh, okay. I think
the Virology Unit.
Yeah, because it became
the MRC Virology Unit. That's
okay.
I was on the university side, as it was a bit separate then. I guess hence my hesitation there. So, but it was the MRC Virology Unit.
Okay, and that was with Richard Elliott, I believe.
Yes.
Looking at Bunya viruses.
Correct.
Okay, I know some things.
Yay!
And then after that, I believe you went to Mount Sinai in New York to work with. I'm not going to pronounce his last name, unfortunately. Palese. And you were working. That's when you started working on influenza A transmission, I believe. Yep, and then from there to Emory.
Yep.
Start leading your own lab where you're looking at influenza A transmission and genome reassortment. Which we'll get into in just a wee second. Because this sort of neatly leads me on to my first sort of question, which involves your work, hence why Anna's a co-host. See, we thought it through. So my understanding of genome reassortment when it comes to influenza A. So influenza A, segmented genome, 8 segments, and viruses, if they co-infect the same cell, you can sort of get swapping of these genome segments.
Yes.
So that, increases diversity within the population. And you can get new progeny that have a different genome from the parental strains.
Exactly. Mm-hmm.
Now Anna's looking at superinfection exclusion, which, from my understanding, again, not a flu scientist, prevents co-infection happening on a cellular level. So once one flu gets into the cell, it kind of pulls the drawbridge up behind it, and then the second virus can't get into that same cell. Mm-hmm. So my question, as not an expert in the field, is I think that the first one that many people might come to, which is, so how can both be happening if you've got this mixing that's happening that we know is happening, and we've also got this super infection exclusion happening? Yeah. So over to the experts.
All right, very good. So I don't know if you want to comment on this, Anna.
You can start if you want, and then I will.
So it's all about timing is my answer, right? Yeah. So superinfection exclusion is very real and very important, and does indeed limit the opportunity for two viruses to operate in the same cell. Mm-hmm. But it kicks in maybe 4 or 6 hours after the initial virus has infected, right? So you have that time window during which additional viral genomes can be delivered to the same cell.
Right, okay.
Yeah, basically, yeah. And my work specifically was about how that works spatially, and the different separate areas of infection interacting with each other. And we see that because of this timing gap, there is actually a small area where they can co-infect together. And we don't know how many cells need to be co-infected with different strains to generate robust reassortment, lots of reassorted viruses, but it does happen despite superinfection exclusion. So because it does with enough chickens, it can. It is the timing and the spatial impact of superinfection exclusion on reassortment wasn't previously kind of well studied, but it does happen, and therefore robust reassortment can be supported despite superinfection exclusion. Yeah. The bit that I would like to ask you about maybe is the influenza virus particularly relies a lot on lots of viruses coming into the same cell, co-infection, to beat the immune system, to get going, to complement each other between semi-infectious virus. So I wanted to ask if you imagine superinfection exclusion as a mechanism that's driven by the virus, how would that work, considering how much of the influenza virus population relies on co-infection?
Yeah, so I think it's useful to sort of separate two concepts here before we answer that question. So one is co-infection between two distinct strains, which is of course the interesting type of co-infection when you're thinking about reassortment, right? Because you'll only get progeny that differ from the parents if you have two different viruses coming in together, right? So that situation is probably relatively rare for the reasons that your work develops, Anna, that you have temporal and spatial differences when there's two different strains in a host that would preclude co-infection between them. Often, not always. But for a separate type of co-infection would be co-infection between identical viruses or very similar viruses, right? That is multiple genomes going into the same cell. They may not be different from each other. Okay, so it's going to be uninteresting from a reassortment point of view, but could be critical for simple propagation of viruses, right? So as Anna's identified, it is sort of a core feature of influenza virus infection that if there are multiple genomes delivered to the same cell, the viral life cycle is much more likely to proceed to completion and to progeny production, right? But that need could be met through this homologous co-infection where there's actually no genetic differences among the viruses, right? So then it's actually really consistent with what you see that within a spatially localized area of infection, you have a high multiplicity of infection because viruses are packed together in that localized area. And yes, they're all basically the same because they're daughter viruses of the same cell, right? But they would complement each other.
Yeah.
They would complete genomes, and they would help each other, as you say, to ramp up infection quickly and things like this. So they would meet that need for co-infection that's so important just for propagation, not for genetic diversity.
Okay.
So the two different ways of looking at the
it's not different, but I do tend to use the word multiple infection just so as not to say co-infection, because when you say co-infection, people really think about two different viruses.
I've
but
had
some
problem trying to explain
creative
this.
terminology is needed
every time. But I guess as well the influenza virus semi-infectious particle problem with transmission, which you also study. If 90% of the particles that influenza virus produces requires multiple infection in order to get going, but the transmission bottleneck is so small, and if you think about internal bottlenecks like your paper showed and Andy Mayling's paper showed, that bottleneck is even tighter together. So how would you get around those two different problems?
Reconcile those different dynamics. No, it's a really interesting question. So just to clarify for the listeners.
And me.
Sorry, I went way too deep in.
It's all good.
Every time.
You can learn.
Our data over the last several years now really strongly suggests that something like 90 to 99% of single infections, so infections where you have a single virus entering a cell, fail. They're abortive.
Right, okay.
But the chances of productive infection are greatly enhanced if you have multiple viral genomes entering the cell. And so one reason for this is the need to have all 8 influenza virus gene segments replicated within a cell. We find that in a single infection, usually only a subset of the segments are actually expressed.
Right, okay.
But if you put in more genomes, there's a higher probability you get all 8 segments replicated. All right, so we see, that is, as I said before, a fundamental feature of influenza virus infection that is highly reliant on co-infection for propagation. So then how do you marry that with the idea that transmission seems to be mediated by a single virus? And the evidence for that is that, we see a tight transmission bottleneck in humans and in other contexts with influenza viruses, such that usually there's a single or maybe 2 viral genomes that get established in an individual. And so we're looking at that from a genetic point of view. You have, one genotype basically is predominant or is exclusively present in an infected individual. All right, but there's a lot of ambiguity about the very early stages of transmission, right? So how do we get to the establishment of one genotype? Is it because only one virus particle infected that person? Or could there have been multiple genomes sort of initially infect the respiratory epithelium, and then through stochastic events early on in infection, most of them are lost, and only one predominates and becomes established.
Yeah.
So I think that's an entirely plausible model. It is hard to test because those early stages of infection are so hard to study. Because the virus is it's a low viral burden at that stage.
Yeah.
But I think it's possible. And then also, when you're thinking about viruses spreading through the air, infecting a person, what are the chances of two of them landing in the same cell to mediate co-infection is also very relevant and sort of puzzle to think about here. And I would suggest that it could be the case that you have multiple virus particles packaged into a single aerosol particle, a single droplet, if you will, which would then readily mediate delivery to a cell of multiple particles. And it's really consistent with what's seen for many different viruses, that you have what's called en-bloc transmission, or sometimes it's called collective dissemination. For example, with enteroviruses, reoviruses and so forth, that they're scaffolding onto biopolymers, or they're being packaged into a vesicle or something like this. We have different ways that multiple virions can be coordinatedly delivered in a coordinated way to the same cell, right? And so we could have that happening with respiratory viruses inside of the aerosol particle.
Yeah.
It's entirely speculative because,
so
again, aerosols are hard to study. And so we don't know how many aerosols, how many virus particles are typically within an aerosol. But I would love to know.
Me too.
It's a fertile ground for further research.
I had related, I guess. I always wondered, again, impossible to study. All of these things are impossible to study. But
just now impossible. That's,
never
we'll
say
get
never.
there.
Yeah, exactly. I wondered whether the 90 to 99% semi-infectious statistic, your paper on collective infection mentioned that it's important earlier in infection to get going to beat the adaptive or innate immune systems. But I always wondered whether it's something that happens later on, like the way that you get an accumulation of DBGs, because then that relates to transmission that we expect happens kind of later on after an infection has been established for a while and there's high viral titers in a person. I always wondered how the timings between transmission and the accumulation of semi-infectious particles and defective viral genomes correlate with each other.
Yeah.
Impossible to study. Very interesting. I would love to donate another PhD to doing this.
Just to confirm my understanding, there is the idea that as the infection continues within the person, their viral titer will increase. So you've got loads of influenza A, but the proportion of that will be semi-infectious particles or DBGs. Is that so? And that might be. Sorry, just to confirm my own understanding, and for those listening.
Potentially. So I think the evidence that we have right now seems to suggest that the 90% semi-infectious particle ratio is kind of established and maintained. I don't think there's been any proof that I've seen that the proportion of semi-infectious particles go up. There is, of course, a dynamic with defective viral genomes and fully infectious virus, where it's kind of a hunter-prey system and they kind of oscillate between. But I don't think that's been shown for semi-infectious particles. But I would love to, if that does happen, how that works with the timing of transmission events is something that I'd love to know.
Yeah, so there's some, I think, different scientists interpret semi-infectious particles differently.
Yeah, that's a good point to make, yes.
I would say we don't have direct evidence that these are a distinct class of particles, and I actually don't use the term semi-infectious particles for that reason. I don't want to imply that is a subpopulation of particles. Rather, it's in my mind an outcome of infection potentially with standard particles. That early in infection segments fail to be replicated, giving rise to an incomplete viral genome within the cell. So it may have been complete in the particle, right? And then in line with that thinking, we wouldn't expect their rates to fluctuate over time during the course of infection. It's just sort of probably a fairly consistent rate of stochastic loss of segments early in infection. would be the model that I think is most plausible. But there is, I think, still some uncertainty around how high fidelity is influenza virus packaging, right? And I do think there are uncertainties there that leave the door open to indeed the particles missing segments, and that being a subpopulation of particles that are missing segments. But I think it's just not clear.
Yeah, it's not clear. I think the failure to produce to express the genome segment, I always wondered whether that was the bit that starts to break down as the infection gets going. It gets looser and looser, or if you're copying from progeny genomes, then you establish a higher and higher error rate, that kind of thing. Yeah. But
well, we haven't tested it. We haven't looked for that sort of change over time.
Yeah. But it's something that's been tickling my brain for the last couple of years. But yeah.
So, just to confirm, I just want to reiterate from my own understanding. So, when I think of semi-infectious particle, I think of something that has the ability to get into the cell, but then not to replicate efficiently, so they come back out. Defective viral genome is something that's out, but with missing parts of the genome. Is that the same for flu, or?
So a defective viral genome, the term is used in the influenza field in particular to describe a genome that has a large internal deletion in one or more segments. Okay. So it presumably had 8 segments, but at least one of them would not be functional because most of the open reading frame has been deleted.
Okay.
But it retains the packaging signal, so there is still a segment.
So it still gets put into the virion, but it's just missing this mid. Okay.
So those arise very commonly in infection and are definitely a feature, but they're quite different from the semi-infectious particles in that when we're talking about a semi-infectious particle, or as I say, I use the term incomplete viral genome to try and avoid referring to a particle per se. But they're, an entire segment or multiple segments are missing. Okay?
Right,
it's not there's a defective component.
Yeah, okay.
It's a missing component.
So it's segments 2, 4, and 6, but none of the rest of them.
Right.
Rather than you've got all 8, but chunks are missing out of the genome. Okay, there we go. We've learned something. Okay, so you're also looking at... I believe the other work you're looking at, so that was to do with reassortment kind of thing. The other thing you're looking at is the transmission. And you're looking at transmission between, I think it's guinea pigs used as an animal model.
That's right.
And I think, because we had a discussion yesterday with, so in doing my homework, I met with Anna, Cal, Bentley, Abbott, and Daniel Weir, shout out to you guys, who are also part of Ed Hutchinson's group yesterday, to have a brief discussion about kind of your work and so I had a bit of a background as to what was happening.
We hosted a little mini journal club. It was really cute.
Club. Yeah, it was really cute. I learned a lot. And I was like, right, okay, so I took all my notes. And I think maybe you've discussed this with Cal earlier, I'm not too sure, but it's an interesting question that Cal brought up, looking at, and Anna, you'll have to fill in some blanks for me here because it's not 100% clear in my mind. I was looking at basically the difference between infection in swine populations and in guinea pig populations and ferret populations. Can you remember exactly what Cal was talking about in that paper? Because it was really interesting looking at the differential between the upper and the lower respiratory tract, I believe it was. Yeah.
So there's been some recent work looking at kind of intra-organ bottlenecks of viral evolution. But your paper specifically, Anice, Cal was talking about the different models and the extent of reassortment between different models and what would be the best model to look at reassortment or transmission. Every model has its drawbacks, but you have to choose the best one. And the difference between using a native host and the kind of lab adapted, mice don't sneeze kind of model. So yeah.
Which is a fact I learned yesterday, that mice don't sneeze. So
or get
if
fever.
Nothing else, which again, very
no
odd.
Fever in mice.
Anyway, sorry. Continue with your question.
So that. I think we had a bit of a discussion about that.
Yeah, so I could talk a bit about the host differences. And so that paper didn't focus on transmission, although we use guinea pigs because they transmit influenza. That's of the reason that we developed that model. But the paper you're referring to, we quantified reassortment, so genetic exchange again, in a handful of mammalian hosts. Okay, so guinea pigs and ferrets were the two model systems, and then swine, of course, are a natural host for influenza. And very interesting when we think about reassortment, because we do see a lot of reassortment in the field with swine influenza A viruses. A lot of diversity generated through that mode in swine flu. Okay. So what we found was in the guinea pig upper respiratory tract, lots of reassortment. We've seen that repeatedly over the years. We just get a ton of reassortment in that system. Also in the ferret upper respiratory tract, lots of reassortment. Okay, and so what that means is that co-infection is frequently happening in vivo, and the genetic exchange is frequently happening in vivo. And so that is important to help us understand, what is the frequency with which viruses access the same cell in an intact animal host. You might have expected that it would be very rare just due to simple numbers, right? There are a lot of potential target cells. And so what are the chances of two viruses coming together in one cell? Okay, so we do see a lot of reassortment in those two situations, the upper respiratory tract of guinea pigs and ferrets. In the lower respiratory tract of ferrets, there was much less reassortment. Okay, and we didn't look in the lower respiratory tract of guinea pigs, so we just don't have that information. But so in lung samples from ferrets, we saw less reassortment, and then also in pigs, both in the upper and lower respiratory tract, it was consistently the case that we could detect reassortments. It's just that the diversity generated through reassortment wasn't very high. Okay. So clearly there are differences among the three hosts, and then also between tissue sites within the ferret in particular. It's actually suggested to us by one of the peer reviewers, and I think it's a reasonable comment. Could this just be about sort of the surface area, the size of the tissue or the size of the animal? And so that is now built into the discussion of the paper, and we just have sort of little data to go on to support this argument, but I think it's a reasonable model that the nasal tract is sort of confined spatially and maybe therefore supports more co-infection and more viral mixing than the lung. Also, pigs are big. and even maybe when you're thinking about the nasal site, it's a relatively large surface area, which could allow just more dispersed infection and not so localized.
That kind of like in a local area would reduce the MOI kind of thing. Is that kind of the concept behind it? Like reduce in a local space?
Exactly. I mean, there's two parts of MOI. One is the number of viruses, and the other is the number of cells, right? So if you increase the number of potential target cells, you will lower the MOI. Okay. There's a lot of other differences between ferrets and pigs that might come into play, including issues related to the innate response, for example. One idea. We don't really know, so that's just one idea.
Yeah.
Interesting. So, welcome back to the CVR, or welcome back to Glasgow, I should say, because when you were first here, it was Church Street, the Virology Unit, as we've discussed. Yep. So what do you think of the new CVR? Is this your first time here, or? It's
my first time to the new facility. It's excellent. Yeah, I'm really impressed.
The big gold cube.
The big gold cube.
The exterior is interesting.
Didn't they said it would like rust and like blend into the surroundings?
It was meant to patina.
It's been 8 years.
Yeah,
not a single. It's bright gold. It's as bright gold as when they installed it.
It's a choice. I remember speaking of Ed Hutchinson, he kind of coordinated our first day as PhD students, and he literally emailed us and said, "Look for the gold cube. That's where you're meant to be." And then I showed up on campus for the first time and was like, "Oh, legit, it's a gold cube." And I was like, "Okay, cool, great." So if you don't mind, I'm going to ask you a few questions just about your kind of how you got to Emory, how you got to where you are just now, and sort of if you wouldn't mind taking us along that journey. So as mentioned in your kind of introduction, you started at the University of Alberta doing biochemistry, and then you made the jump to Glasgow to do virology. So kind of my first question is, what inspired the jump to Glasgow, of all places? And then the next one, which might be a bit more informative, and I'm from Glasgow, I can say that.
Yeah, you can say that.
And the other thing is, what made you want to make the switch from biochem to virology, if there was an impetus behind that?
Yeah, absolutely. So I mean, biochemistry is very broad, right? There's a lot of biology that falls into that field. So my undergrad degree didn't sort of point me in a particular direction early on, except that I think it was in my third year at the University of Alberta, I did take a virology class, and I loved it. It was just really compelling. I spent hours in the library reading Field's virology. I was very into it. So that is how I chose virology, was through that course. And I think as an undergrad, actually, it was well, I guess a bit discouraged by the complexity of biology and sort of this concern that we would never understand the inner workings of a cell. It was just too complex. And so I discovered virology in that moment, my own personal journey. And viruses were very appealing because they're simple, relatively simple, right? And so I think that's sort of what really captured me. And then how did I get to Glasgow? It was quite a leap from Edmonton, Alberta. So I did really want to work on a relatively simple virus and therefore an RNA virus, with a small genome, a few proteins encoded. That's what I was looking for. And in Canada at that time, 20, 25 years ago, there wasn't a lot of RNA virology going on. Okay, so Canada has a pretty small virology community, and the sort of main features were pox viruses and herpes viruses. So, they're big, complex viruses.
Do you know why there was a particular emphasis on DNA virology in Canada?
I'm not sure that it was by design at all. I think it's one of these things where you have sort of a prominent virologist within a relatively small community who trains all the students and postdocs, and then they go out and sort of populate the virology community at other institutions also. And so they tend to have our latest focus areas.
That must have been interesting for, skipping ahead a bit, moving to the CVR and having lots of, well, not the CVR at the time, but...
For sake of conversation.
The CVR, if I say CVR, I mean either or. Where people were working on lots of different viruses and a bit of a richer diversity of community, I guess.
Right. So I think, as a 22-year-old, I didn't sort of recognize what I was choosing to be an excellent choice. I don't think
any of us were. I definitely didn't.
But I was very lucky, I think, in that, I went to a virology institute. I didn't go to, one lab in whatever, a biochemistry department, for example, that happened to do virology. I went to an entire institute that was focused on virology. And it's a beautiful thing because... you then have a lot of scientists, a lot of creative minds, all sort of thinking in that similar space, and it was really awesome. So I looked outside of Canada because I wasn't seeing what I was interested in for a PhD there. And to be honest, the American system was just a bit foreign to me in terms of the timeline needed for application to grad school and things like that. And so I looked to the UK for that reason. I had never been to the UK before I moved here for my PhD.
We love
right in at the deep end.
I'd never been to Scotland, but at least I was like, from the UK.
But I found Glasgow. Honestly, there was a virologist in Edmonton that recommended it to me, and also by looking at the Journal of Virology, there were a lot of papers from Glasgow. There's something going on in Glasgow. And so I emailed Richard Elliott, my eventual PhD advisor, to ask whether I could come, and he was interested right away. And I now understand, after having been in his lab for a few years, that he was actually really open to bringing people from other countries. It was something he sort of always strove to do to have a diverse group in the lab. And so I expect that's why he entertained my email from the beginning. That he kind of liked that idea. So Richard really facilitated getting me here. Yeah.
Oh, amazing. Now, I do have to ask the question, how was the accent when you first moved? How was understanding people?
The Glaswegian accent.
Coming to Scotland was really fun because it was, it's quite distinct from home. There's a lot of cultural differences, but actually there's a lot of similarities too, right?
Okay.
Because there's a big Scottish influence in Canada still. And so bagpipes are all the parades, for example.
Nova Scotia.
Nova Scotia.
Yeah. My paternal grandmother's family is Scottish.
Okay.
So there are a lot of connections, a lot of cultural similarities that are maintained today. Okay, so there were some similarities, including the language to an extent, right? So of course I could communicate right away, but there are all these sort of funny differences. Like my first roommate, the first day I met her, she asked me how I had my tea. I'm like, tea? I drink tea sometimes, but of course she was wanting to know whether I'd eaten my main meal that day. So just things like that happened a lot. And I never got to understand, for example, like the taxi drivers and so forth.
I was literally, my next question was taxi drivers, because they, if you don't know, Glaswegian taxi drivers have very strong accents and will talk to you the entire journey. As a general rule of thumb. maybe one day we'll get there.
Yeah,
we'll do some lessons or something.
I think for me the culture shock was the hospital. Like I took my roommate into A&E and the people chatting to me, I had no idea. I was just like, yeah, yeah, it's good. And then we had handymen in, they were like, oh, this is really important with the boiler. Nothing else. So I'm so glad that we moved out of that flat before it exploded, because there was something important that we needed to do and had no idea. But yeah, I guess changing full continents and that language barrier.
Well, yeah.
It was a bit of an adventure.
But yeah, you got through it. You did your PhD with Richard Elliott, and then you went off to Mount Sinai under Peter Polese to look at influenza A transmission. Now, was that your first postdoc?
Yes.
Okay.
My one and only.
And okay. And kind of, because I've done something similar myself. My undergrad was focused on mycology, and then I've moved on to virology. Yep. But for me, the overarching theme has always been proteins. That's the one thing that connects. That's what I study. So kind of, what was your kind of train of thought when you were thinking, okay, I'm looking at postdocs, I'm doing all this work on bunyavirus, and Peter Polese has this lab over in Mount Sinai doing influenza A. Let's talk to him and see what happens. Kind of, what was that train of thought for you?
Sure. So working on bunyaviruses in the early 2000s is a small field, and the literature was not super deep, right? So if for example, I was working on genome packaging, and so if I wanted to read around that topic, the obvious place to go would be the flu literature. So influenza viruses and bunyaviruses have in common that they are RNA genomes in a negative sense, and they're segmented. So bunyaviruses have 3 segments, and influenza viruses have 8 segments. There's a lot of similarities in the genome structure, and therefore, and sort of the processes that play out during the viral life cycle in the cell. So I was pretty familiar with influenza for that reason, and it was also sort of, I guess, a literature I admired, in that it was further developed than the bunyavirus literature at that time. And so in looking where to go next, that was quite attractive, right? To move into a field that was larger, as a greater critical mass, I guess a higher level of public interest and understanding of influenza being a problem also. So yeah, that's sort of how I set on influenza in particular. And then Peter Polese is really a giant in the field, and so it was an obvious choice if I wanted to go to a postdoc that would give me a chance to really get established and build a name for myself. He was also the postdoc advisor of Richard Elliott. So there was that connection sort of through the scientific generations, if you will, of Richard had come through Peter's lab, and so that was an example, if you will.
Okay, cool. So you go from Scotland to New York. I don't know anything about New York, so I can't comment about the taxi drivers, sorry. But how do you find your time postdocing in New York? What was kind of like the main difference for you between your PhD experience and your first postdoc experience, and kind of how did you handle that change?
All right. So, one similarity actually between the CVR and Mount Sinai, the microbiology department at Mount Sinai, is that both are virology institutes, if you will, right? So although it's called the Department of Microbiology at Mount Sinai, there isn't any bacteriology, it's all virology.
Okay.
And so we have labs working on a number of different viruses, and it's just a really sort of cohesive, active place where everybody's very engaged. And so, again, it was great to go to such a vibrant virology community. And then, it was different. New York City in general is different in that it's very intense, right? And so Mount Sinai was a microcosm of that. The beautiful thing about it is everybody's very engaged in their science, very motivated, excited to hear about your work, and you get a lot of discussion and things like that. But it is also an intense environment, right? Where it's sort of like there's not a lot of chance to step away from it, basically.
Yeah, I think we've all heard about American postdocing. So it seems a bit extreme.
I wanted to know what the roles were. Did you feel a distinct role between PhD student and postdoc? Or do you think that was just the environment, or do you think it's built into the job?
I think more the environment, actually. I think that of course, a student is very green and is learning, and it needs to be given a little more time and space to do that. Whereas a postdoc, brings their own expertise to the position, which may be a little different from the PI also, right? So that can be quite valuable. And a postdoc usually will hit the ground running a little more than a student because they have that experience. But the main job of both a PhD student and a postdoc is to advance a research project, to generate data and to publish it. So in that way, the role is the same. And I think that was true here in Glasgow and also Mount Sinai, that the students and postdocs were all kind of in the mix together. Right. I would say that, although I describe my postdoc as very intense, and I do think it's in part a feature of New York City and in part a feature of that department. It's not the case across America. It's like not homogeneous in this way. There are American postdocs that are, I guess, more conducive to having a balanced lifestyle, if you will.
Okay.
So I don't want to mislead
Some are
anyone
harder
there.
than others. Yeah, well, more intense, than others.
Turns out you should avoid sweeping generalizations. That's that's a good skill for science, just in general. So you've made this transition. You're now postdocing in Mount Sinai. This is something we've vaguely had chats about it within the CVR. Is kind of there was this internal seminar type thing that we did basically on applying for fellowships and starting your own lab and things like that. And one of the things that I think I can't remember exactly who said it, unfortunately, but someone here said that you'll know when it's time to start your own lab or when you want to start making those moves. And was there anything for you like that? Was there like a light bulb moment where you were like, oh, actually, I've got this idea. Yeah. Let's try and pursue it, or when do you start thinking, wait a second, I might want to...
Yeah, and how early did you know that you wanted a full career in academic research, I guess?
Yeah.
That's two
questions. Both good questions. I'm going to start with Anna's.
Okay.
So, as an early PhD student, I really didn't have designs on an academic career, and I think I didn't know what I wanted. I hadn't really thought about it, and I didn't have the confidence to assume that I could direct a lab. Just the sort of part of that involves directing people, I didn't feel I was well placed to do.
Yeah. Well, as a PhD student, you're already fighting like scientific imposter syndrome, let alone like management. Like that's too far away for me to. I need to focus on my Western blot.
So definitely that was me, and it was a progression during my PhD years of sort of seeing myself as a scientist and understanding that actually I did have that potential to carry on in academics and direct a lab, not just sort of work in a lab. So I think with the help of Richard, my PhD advisor, sort of came to that realization as a student that yes, this was something I could do. I had the sort of talents that would allow that. And so Richard was actually really helpful in just sort of pointing out to me that I should be more confident in my work and not to shy away from that, basically. And then the postdoc years were also very formative in that Peter Palese sets very high expectations for everyone. Okay? And that is sort of a major feature of his mentorship style, and it's actually sort of a very effective style. Okay, so he sets extremely high expectations, and then people reach them. Okay, so I was very productive in my postdoc, and that's really important for your trajectory to go onward into a faculty position. Now, in terms of readiness, so. I think in the United States there's definitely sort of a rhythm where it's usually going to be around 5 years into your postdoc that you would move on to a faculty position. But there is quite a sort of well-established cycle or tempo there where you postdoc for 5, maybe 6 years in our field. That's a typical length anyway. And then you go with on the market, they call it, right? You sort of tour around different universities, interviewing for faculty positions. And then if you land one of these positions, you're given a bolus of money. That's called a startup package, and a laboratory space, which probably has room for about 6 people. Okay, it's pretty big. And your career is launched in this way, where the institution gives you support to get started. The money would usually last about 3-4 years. Okay, so in other words, you're not looking for a fellowship like like you are here.
Yeah,
it's
that's
quite
a lot more
distinct.
formal of
Yeah.
a process, I guess.
And yeah, it's a much starker transition from a postdoc to a faculty position. It's night and day. And having landed a faculty position, you're given this big push of funding and support. Whereas here, it's a lot more gradual transition, right? From trainee to lab director. But there is definitely a concept which is touted pretty commonly in the United States about readiness, right? When is a postdoc ready to make that transition? So yes, it's typically 5 years in, it seems, but there's also, of course, you need to build your CV, you have sort of a portfolio. And you do need to have ideas, okay? And I think that's not something you sort of wake up one day and you have ideas, right? It is sort of a learned skill, okay? And so I think that's important for students to understand, actually, that, at your stage, it's not unusual to sort of not have a lot of ideas in terms of like testable, impactful hypotheses. That is something that you get good at over time as you get to be more familiar with your field, as you read more deeply, and as you sort of understand what are opportunities that would actually be feasible to go after in the laboratory, given your tools and so forth. Right? So I think that's a major point of readiness, actually, as a postdoc, is to, and it's also a skill set that develops over time. I think I've gotten a lot better at it over the years in my current position. But as a postdoc, you do want to have a couple of good ideas that are different from what other people are doing, so it's something to start your own program on. And so that's definitely important. Yeah. There are other factors of readiness that just you don't acquire the skills until you're in the job, I think. And they are the things like managing people and budgets and so forth. You just got to learn on the job some of that.
Well, I mean, you learn on the job from day dot and
everything.
PhD students.
Because it's more formal, like that position and that space comes with training, I assume, from the university and help with that kind of thing. No?
Well, not very much. So for
it's just like sick.
You're smart.
You're smart. Here's some money. Here's some space. Do it yourself.
No, you don't get training on how to hire personnel, how to fire personnel, how to manage personnel. You don't get training on that, and you should. There is now more and more training for the mentors of PhD students, right? So there's sort of these workshops and whatnot around mentoring PhD students. But in terms of, just managing a laboratory staff, a technician and so forth, there's potentially room for some training in that, which doesn't exist to my knowledge. Similarly, for the financial piece, there's no training. There is help. You do, there are financial analysts that support scientists by keeping track of spending and things like that. But it, maybe to have an intermediary that could interpret what they're saying, so really know what's going on.
I was gonna say you have to keep track of suddenly you have to keep track of everything. But I was gonna ask, like, because you're launched with money, at what point do you transition to writing grants?
Yeah. So. As early as possible. Okay, so many new faculty members will write grants, R01 grants, so they're major grants, within their first year there in the position. I think I may have submitted my first one maybe around the 1 or 1.5 year mark. Okay, and what I was doing in that year and a half was collecting data, preliminary data, which is a really important piece of an R01 grant going to the National Institutes of Health. So that's sort of well understood among scientists in the United States that you can't just put in your ideas. They do need to be supported by some preliminary data that would sort of give the rationale for your hypothesis potentially, or indicate that the assays or screens or whatever that you're proposing are feasible. So to get to the point where I was ready to submit a grant, it took me about a year of working at the bench to generate data.
Are you still based at the bench a lot, or have you completely separated from it now?
I'm completely at my computer now.
Did you was it a slow separation away, or there was a point where you went back, you left and you never went back in? Gone forever.
I came to that point eventually, but
for context,
so
I have Ed's pipettes, I have Ed's TC shelf. Like, he left one day and I just took over and he never came back.
One plaque assay too many.
I guess that was the point where I didn't come back. I gave my bench to a student. And there was nowhere to sit anymore.
Yeah, exactly.
But no, I functioned as my own postdoc for about 5 years. So I mean, it's hard to recruit postdocs when you're a really new lab. Is one reason for that. But also, by the time you start your own lab, you're a very well trained scientist and you're an effective experimentalist.
You can do it quickly.
It can be quite productive to just get in the lab and do some experiments yourself. So I kind of juggled the two things for about 5 years, and then transitioned out of the lab over the period of a year or so. I think. And there does come a point where, well, for me, I was really distracted by things that would be going on in my office, on my computer, or whatever it was, talking to collaborators, talking with students, things like that. I would maybe set up an experiment, a transfection in the hood, and there was a 20-minute incubation, so I go to my office and start doing emails, and then I forget all about the experiment until the next day.
The cells are dead. It's
fine. There just came a point where I wasn't a very good lab worker anymore because I wasn't able to focus on it.
Yeah, your job transitioned away from it. And I think it was interesting during the pandemic when you saw some PIs step back into the lab because there was people couldn't come in. I thought that was very cool, but that was just a comment.
I love bench work, and it'd be fun to go back. But I also love my current role. I don't miss it because I still get to think a lot about science, which is the most fun part of doing
my
science.
undergrad supervisor we would work in the bench during the day, and then during the night he would come in and do it, and then you'd come in the morning and all of his stuff was like rearranged. It was like having a ghost worker in the lab. It was strange. Yeah, like a secret Batman night lab worker.
Secret science is a thing.
Exactly.
It does happen. So the next question I just have about this sort of your career trajectory, before we move on to if you have any advice for early career researchers, and it's, was there any obstacles that you faced personally when you're on that upward trajectory from PhD student to PI, and kind of, do you have any advice about how you overcame those obstacles that might be pertinent?
Yeah, I mean, I guess science is hard, right? So without sort of naming a specific event, there's definitely the challenge of just keeping going in it, right? Your experiment has failed 6 times, and here you are doing it again. It can be very frustrating. And then also, I think in academic science, there's always a lot of uncertainty also about what your trajectory is, right? Because not everyone goes on to a faculty position, and there's other paths to take, but you maybe don't know which path will be yours from a very early point in your career. And so to stay motivated through the slog of experimental research, given that uncertainty of the career trajectory, I think is hard. And I guess what keeps many of us going through that is the joy of discovery, right? If that experiment does work, well, it's very rewarding. It's very exciting, right? And it's like a drug, we keep coming back for more.
You get a dopamine hit every 6 months, and you're like, oh, this is why I did this. And then you're back in. The highs and lows are crazy.
Oh, yeah. I was literally about to say I've just finished a block of experiments and just got them all to work, and I'm like,
congratulations.
Right, now I've got the data analysis to do, so slightly marred, but 100%. And it's so funny that that's from every level, from PhD to postdoc to PI. Is that highs and lows? You still got to ride the roller coaster.
So it is still present in my current reality. I would say it's a lot easier now, that part of science, because there's 10 people in my lab working away on projects that I am interested in and heavily invested in, right? And if 3 of those 10 projects are going slowly, well, maybe 7 of them are more interesting, right? So I have a hand in a lot more different things, and that just sort of avoids this issue of getting into the doldrums.
You've diversified your supply of the dopamine.
Well, basically, yes. Right.
What's really I don't know what this says about us as people, but I said you've outsourced the disappointment is where I went to in my So I don't know what that says about us, but let's not think about that too much.
But I could also see the longer arc of it easily now too,
absolutely.
right? So even those projects that were really hard for a period, and it may have been a period of years, to be honest, right? They do tend to come to fruition. You know, every student graduates, and they usually have a beautiful dissertation at the end of it, right? And so to see that repeatedly, that, you know, the challenges do turn into successes, it is very encouraging. Yeah.
Maybe it's nice to see where the projects fit in together as well. Like
Also you can
start to...
you can build up sort of a portfolio of research that, you know, you sort of have some ownership over, and that's very fulfilling. Yeah. You know, it's not just one paper, but it's a story of, you know, maybe 5-6 papers come together to tell the whole story. And so to have the time in your science to do that, you know, to be in that same position for actually decades is a beautiful thing.
It must be very satisfying.
Yeah.
I think it would, because it's just like, it's like little puzzle pieces coming into place, right? And then you're the one seeing the whole thing. Like,
that sounds really cool.
It does sound really cool.
Maybe.
Well, this sort of leads me on to my next sort of question. So I'm going to ask you some advice for some early career researchers, so PhD students, people in their first couple of postdocs. Now, this is more of a personal thing, just for me now. So I do kind of know where I want to be in 5-10 years' time, but I struggle somewhat with focusing on the bigger picture when you're dealing with the day-to-day bench work. I've got a Western blot to do, I've got transfection to do, I've got data to analyze, How did you find that? How do you find focusing on the bigger picture and saying, right, okay, here's my goals for, you know, 5 years' time, 10 years' time, whatever it may be, whilst also keeping yourself doing the day-to-day and getting the job done? Because I find like sometimes I struggle to think of the next step because I'm so focused on this step and making the Western blot right and making it work and
Yeah.
that you kind of fix. So any advice for balancing those two?
Yeah, that's a great question. So I mean, you have to focus on the day-to-day or you won't get it done, right? And you're not going to be productive if you don't do the Western plots. So, you know, you have to sort of be focused on the details, definitely. But then I think to periodically step back and remind yourself that the bigger picture is really important, right? And I think the way it's sort of naturally built into the academic life cycle actually is, you know, you need to write up the paper at some point. Or maybe you're going to give a seminar, a presentation. Maybe it's just a lab meeting. You know, it doesn't need to be a sort of big deal. But putting together a slide deck on your science forces you to think about, you know, what was the original question I'm going after? What's the progress I've made towards it? And at a pretty deep level, because you have to explain it to people who maybe aren't familiar with the project, right? And so it forces you to really step back and think about, you know, what are the important issues here? Such that you can articulate them for others. So I think it is those moments that still help me to, you know, look at the big picture and not just focus on each individual experiment. And so that's, you know, about the science progressing, not so much about the career path. I have to say I am not the best example of a person who had sort of a career goal in mind and then went after it year on year.
Okay.
I have really followed my nose in terms of my career path, and you know, it's worked well for me. But other people are much more planners and sort of.
I think it's valuable to hear both. Like
absolutely.
My Ed Hutchinson, my supervisor, he was like, "Yep, I wanted to be a PI, so I did this."
Right.
And I was like, I was like, "That's a lot of pressure, Ed. Like, I don't know what I want to do." So it's nice to hear it a little bit more.
Sure. I've always just done what I was interested in, really.
Yeah.
I guess fortunate to have the freedom to have done, you know, do that, right? You know, get up and move to Glasgow from home because there was an interesting project that I wanted to pursue. You know, not everybody can leave their family behind like that, for example. So I have been very fortunate to have that flexibility to just go after something that I deemed interesting. But it's definitely the way I've operated. You know, I didn't do a lot of interviews at the PhD stage. I didn't do a lot of interviews at the postdoc stage either. I just sort of found a place that looked interesting and went. And, you know, I see my students graduating and really spending a lot of time thinking about what to do next and you know, interviewing in multiple different places for postdocs and things like that. And you know, I do wonder if they're overthinking it sometimes. I think different personalities handle it differently.
I think it's a balance, definitely. And but it's nice to know that you can follow your curiosity. Just keep the curiosity at the center, and then you will find a way to doing something that's cool.
Path.
Yeah, you'll find a path.
There's no one path.
No, exactly. Yeah, exactly.
And one last thing, something that Anna, you brought up earlier on, imposter syndrome.
Ooh.
So I think this is, I've just gone into my dreaded, apparently cursed second year of my PhD. Everyone's told me it's cursed.
The second year is cursed. The second year blues is the worst time.
And I have to admit, like I said, I have just got this block of work done, but middle of the work, I was like, okay, I can't use a pipette. What am I doing here? What is a cell? What is a virus? And obviously, that kind of reoccurring conversation you have with yourself of, no, I'm here for a reason, and blah, blah, blah. And, you know, we briefly mentioned it when you're talking about just getting through the slog of experiments not working and coming to the end of these cycles of work. But specifically in terms of imposter syndrome, do you have any, advice or kind of idea on how people can just remind themselves every now and again, actually, no, I'm meant to be here and I'm meant to be doing this work and I deserve to be here and deserve to be doing the work. Yeah.
So it's super common, imposter syndrome, right? I mean, think of a scientist and probably they experience this at some stage of their career also and may do so on an ongoing basis, right? So I think that's the first sort of important thing to recognize is that everybody goes through this, right? And then how to sort of coach yourself out of it. I think my best suggestion is to actually reach outside your comfort zone and force yourself to do that. Put in for a talk at the ASV meeting, something like this that's going to be challenging to you, right? Through success in those situations, it'll really build your confidence, right? But an important piece in that is to also not let failure in those situations sort of really hurt you. And so you have to be somewhat robust to failure and keep trying, right? And so the place that scientists usually fail is in applying for funding, right? But you have to go into that knowing that that's the most likely outcome is that it's going to be rejected. Okay, and often papers, especially if you shoot for higher journals from the beginning, you're going to go through a series of rejections. Okay, but I think it's a useful process to go through actually. Probably at the end of the day, your paper will be accepted somewhere really quite good, and you'll be very proud of it. And so then those early rejections don't matter anymore. Going through that experience sort of a few times, you really sort of figure out, well, this is part of it, and actually look at it very successful. Right? It's very nice work. Right? I think stepping outside your comfort zone, not being afraid to fail, you will sometimes succeed, and then that will reinforce to yourself, I do belong here, and I'm doing the thing.
A bit of a personal question, feel free not to answer, but do you still experience imposter syndrome, or has it changed, or...?
I think in some contexts, I guess, if I'm stepping outside my comfort zone, if you will, right? So 12 years in, I'm very comfortable in my faculty role and directing my lab and doing my science. I think something that I'm still sort of growing in professionally is reaching beyond what I do in the lab, right? Viral genetics type work. Through collaborations and into sort of related fields and sort of working with scientists in other fields and pursuing science that I don't feel is mine, and it is super important, right? These collaborations, and it's a way that scientists can have a bigger impact is by working with others in just a bigger space. So I guess that's sort of an area of growth for me, and anywhere where you're stretching yourself like that, it's going to happen. Some imposter syndrome.
Yeah, no, definitely.
And I think something I picked up from your advice there was it's kind of the more opportunities you have, obviously, taking my example, I'm in second year of my PhD, you're coming up to your final year. The number of opportunities you have to go out there and fail or succeed, as we hope we all do, but is less. And so you're kind of, it's more of a sting because it's your first time getting a paper rejected, it's your first, and then eventually it's like, oh, I've done this 10 times, I've done this 20 times.
Robust to it.
Robust
you become stronger in yourself. Especially if you have a bedrock of work that you've done and it's been recognized. Absolutely. You're like, well, I've got this. Like, this is my chest of positive results, and if there's something in there, then that helps. But I think
just with experience in life, you gain perspective also, right? It's harder when you're young. A big one for me was having children, actually. It's like, I've been through that. I mean, this is easy.
Fair enough. So advice is have children?
Just the harder the thing is, the easier the easier everything else looks. I guess.
Yes.
Do you have any other questions specifically?
Career stuff? Not really. I did want to say though that like forming collaborations with people that aren't in your field, I found that challenging during the pandemic, and when we came out of it, it was much easier. And that was not that anyone's asked, but that was my top tip against imposter syndrome. When you click with someone from a different field, but you relate on the same topic, you're like, wow, I know what I'm talking about. And then you can start to like foster collaborations and feel part of the scientific community. And I feel like that was my main anti-imposter syndrome.
Yeah, good.
Like pill. Cure. Talk to people.
Talk to people and just get out there, put yourself out there, out of your comfort zone. Well, thank you very much for taking the time to talk to us today. Before you go, I believe Ed Hutchinson described me as a well of chaos at one point. So I'm known as being quite a chaotic person. So I've got one last question for you, which, give me 2 seconds. So I'm calling it the cliche in a twist. So you're going to pick a question from here, and you'll understand what's happening in just a wee second. So the first question is a very cliched question that you get all the time. The second part of it is a slight twist to add on to the first part of the question. So if you'd like to read your question for us.
The cliche. If you could have a superpower, which superpower would you want and why?
Very cliched question.
The twist. And if you could have one regular skill or talent you don't currently possess, what would it be and why? Okay.
And just to give you a bit of time to think, my example, because I thought about my examples for all of these, would be I'd want to fly, obvious reasons, transport. One regular skill that I don't have that I'd quite like to be able to do is juggle. Just because I think it's like a fun, easy party trick. You get handed, an apple and orange and a tangerine at a party suddenly.
Yeah, it's
you're the
but fun skill.
Exactly, and it's like low stakes. Like, so that's my kind of example.
If you need, I can try and answer. Is it if you need time to think?
Yes, you go.
Okay.
Good.
I don't know about the twist yet. Superpower, obviously, stop and pause and rewind time. You'd want time control. Absolutely. It's like teleportation and all of that, but like built in. And you can replay like awkward moments and fast forward the really boring ones. If I had one regular skill that I don't currently possess, what would it be? Oh, Lord. Oh gosh, I don't know. I don't actually know that one. We were talking about death dropping. The last
Death
podcast.
dropping. There we go.
A specific dance move that I don't currently possess.
Yeah.
What was the question for that? It was like, what
would you want to be remembered for? Yeah.
And I said, this particular dance move that I can't do, but once I can, it would be amazing. So that.
This is the story arc is complete. We did another podcast, and the arc is now complete.
Parts to get the joke, but yeah.
Yeah, there we go.
Okay, so having just flown here from Atlanta, I am going to go with teleportation for my superpower.
Yep.
So just to jump around the globe would be awesome.
Mm-hmm.
And then the regular skill, coding.
Excellent
Excellent choice.
Yeah.
We can maybe chat about ChatGPT after this, because do we need that skill anymore? Who knows?
I've dabbled
definitely
a little
do.
in it. I'm sort of too far along in my career to learn it properly, and it's just so useful in so many areas.
So good, yeah.
That was perfect. Well, thank you very much for joining in with my little bit of chaos that I liked to introduce at the end, and for talking to us throughout this, I believe, hour or so that we've been talking. Just quickly before you go, is there anything else you'd like to say?
No, thank you. It's been great. I really enjoyed it.
Good. And
Thank you for a good conversation.
First podcast, I believe.
Yes.
Good. First of many, hopefully. And where would you like people to find you? AKA, what's your Twitter handle?
My Twitter handle is @Anice_Lowen. I have not been very active on Twitter since it was taken over by Elon Musk.
That's fair.
That's
totally valid.
You can find our website at lowenlab.com.
Perfect. lowenlab.com. And Anna, where would you like to be found?
I sometimes use Twitter. Sometimes. At Anna Sims Biol on Twitter. BIOL at the end.
Perfect. And, I'm Stephen Devlin. You can find me on Twitter at sdevlinbio, I think it is. Thank you so much for watching, for listening, for engaging, and we will see you next time on Research Goes Viral. Bye!
Bye.